All right, continuing on with our magic school bus ride, let's review. We have looked at the formed elements of the blood. The formed elements of the blood being the blood cells, your red blood cells, and then all of your white blood cells. Know this chart, table 10-2. Okay, that's one thing you want to know. Moving on, let's take a look at how these blood cells are formed. Do you remember from the bone unit, one of the functions of our big bones is blood formation or hematopoiesis. It occurs in the red bone marrow that lives in the medullary cavity of the long bones of your thigh and your ilium. They start out as one common blood cell, a hemocytoblast, and it splits or differentiates into two different types of cells. One cell is a lymphoid stem cell, and that produces your lymphocytes. The other one is your myeloid stem cell, and that produces all other formed elements. So let's look at this. You've got your hemocytoblast here, and you've got your lymphoid stem cell that only produces lymphocytes, nothing else. Your myeloid stem cell produces all the other types of blood cells. Your lymphocytes are 20 to 45% of your white blood cells. They are your B lymphocytes and your T lymphocytes, and they are your they're your first level of defense in your immune system. And the lymphoid cells are the sole producer of these lymphocytes. All others come from your myeloid cells, okay? Your erythrocytes, or red blood cells, platelets, monocytes, neutrophils, eosinophils, and basophils. Try saying that 10 times fast. So, red blood cells have no nucleus. They can't divide, they can't grow, and they can't make proteins. All they do is carry oxygen. They only live for 100 to 120 days. That's it. Remember that. When they are worn out, they are filtered out in the spleen or the liver, either or, by phagocytes. Phagocytes are those cells that go and eat other cells to clean them out of the way. And they are, as they are replaced, or as they are lost, they are replaced by the red bone marrow. You have a negative feedback loop. Ooh, here we go again with the negative feedback loops. Your negative feedback loop is controlling your red blood cell production. So, it's controlled by a hormone called erythropoietin. Your kidneys produce that hormone in response to oxygen levels within the blood. Let's take a look at this. Your homeostasis maintains your even levels of blood oxygen. When your body or your kidneys detect a decreased amount of oxygen in the blood, whether it's due to a decrease in your red blood cells, whether it's a decrease in the amount of hemoglobin within the blood cells, possibly anemia, or it could be decreased amount of oxygen in the atmosphere. Say you go to Flagstaff. That could be a reason why that happens. Your kidney releases erythropoietin to this response, to this decrease in oxygen. The erythropoietin targets your red bone marrow in your long bones to increase the output of your red blood cells. Now, if we go back to looking at how much oxygen the red blood cells carry, it's a lot. 280 million, I think we said. Go back and review that number. And if you're listening to this, here's an Easter egg. Go back, review that number, give me the exact number and a comment on the topic, and I'll give you a point. So, you've got an increase in red blood cells that can carry the oxygen, which in turn increases the amount of oxygen in the blood. Once that happens, it rises back up to normal oxygen levels, which then turns off the erythropoietin production, and then you're restored back into homeostasis. Looking at the formation of your white blood cells and your platelets, they're controlled by CSFs, or colony stimulating factors, and interleukins. They prompt your bone marrow to generate leukocytes, or leukopoietin. And then your thrombocytes are produced by thrombopoietin, which are your what? Your platelets, right? Thrombocytes are your platelets. So thrombopoietin triggers platelet production. Leukopoietin triggers leukocyte production. Remember the sounds. Let's look at hemostasis. Platelets, you need a lot of them. They are by far the largest formed element in your blood, and that is because you need them for hemostasis. Hemostasis is your blood clotting. There's 3 phases to it. You've got your vascular spasms, your platelet plug, and your coagulation, which is the actual blood clotting part of it. Let's look at vascular spasms. It's exactly what it sounds like. You get a cut, your blood vessels spasm to decrease the amount of blood loss. It's kind of like it creates its own little tourniquet without actually having a tourniquet in a smaller level. So you've got your vascular spasm, which has the decrease, the blood vessel, the tunica media contracts to decrease the amount of blood that's lost. The next stage is platelet plug formation. So we've got our constriction of the blood vessel. When you have the blood vessel that's cut, it exposes collagen fibers. What happens is platelets sense it and become sticky to cling to those fibers, kind of like debris in a river sticking to the branches of a tree that may be pushed over in the river. That's kind of what happens here. So you've got these sticky platelets that are on the fibers, the collagen fibers of the exposed blood vessels. These are your anchored platelets, and they release chemicals to pull more platelets into the area. And it's a cascade. It just keeps pulling more and more platelets into the area until ultimately there's a plug or a white thrombus that's formed. Now that we've got the plug preventing the loss of blood from the blood vessel, the third mechanism of this, or the third mechanism of hemostasis, is clotting or coagulation. There's 3 stages of this, and we're going to break it down nice and easy. Stage 1 is the formation of prothrombinase. Stage 2 is the conversion of prothrombin to thrombin. Stage 3 is the conversion of fibrinogen to fibrin. The formation of prothrombinase allows for the conversion of prothrombin into thrombin. The conversion, or the end product of thrombin, then allows for fibrinogen to turn into fibrin. That fibrin is actually what creates the mesh on top of the blood plug. So if we go here, you've got your plug, you've got the cut, you've got the plug on top of it, and then on top of that plug, you've got the formation of fibrin, which is this mesh that then begins to knit the 2 sides of the injured blood vessel together. It's kind of like pulling the sides of an injured teddy bear together with all the stuffing on the inside. And as you're pulling it tighter, the stuffing wants to come out, and you just have to keep pushing it back in. It's a similar thought process in that the fibrin creates this fabric that sits on top that then begins to pull the 2 edges together to create the seal. Once that happens, you've got the blood clot, and it happens specifically within 3 to 6 minutes, and it stays in place until the endothelium is repaired. Once it's repaired, it gets broken down and recycled back through the kidneys into the body. There are times where you have clotting within your blood vessels that you don't necessarily want. That clotting is called a thrombus. It's a clot in an uninjured blood vessel and can be deadly when you find them in your heart or in your brain. You can have pieces that break off of that thrombus and float away in the bloodstream that also can later affect critical vessels in your heart or in your brain. Those are called an embolus. When you have an embolus, which is the piece of thrombus that's broken away, the piece of clot, what they'll do is they will give you an anticoagulant, which is a drug that prevents the clotting factors from forming. Drugs like this are Coumadin or heparin, and they basically prevent the blood clot from forming so that the embolus may be smaller or may avoid being shed. Calcium and vitamin K can help with blood clotting. So there's factors that go both ways in the blood clotting world. Finally, we have bleeding disorders. There are several bleeding disorders. They can be found on page 342 in your textbook. Know that chart on page 342. A couple of the types that we have here is we've got thrombocytopenia, which is you don't have enough platelets within your body. So by not having enough platelets in your body, what happens is you can't create blood clots. So in not being able to create blood clots, what you'll see is blotches on the skin of a person who has this because they simply just don't have enough platelets to stop bleeding. Another version of a bleeding disorder is hemophilia. There's several different types of hemophilia. You can find this, again, page 342 in your textbook. Know it, study it for the test. Let's talk about blood groups. Blood groups and transfusions. When you have a big injury, you may have to have a transfusion. That is, large amounts of blood have been lost. A loss of 15 to 30% of your blood can cause weakness. Only 15 to 30. Could you imagine if you lost 40% of your blood if you had a huge injury that way? That's where blood transfusions come in. Loss of over 30% of your blood can be fatal. It causes shock, and it's potentially a shock that your body cannot recover from. Transfusions are also given for anemia, like if you've got severe anemia or thrombocytopenia. Going back, thrombocytopenia is when you don't have enough platelets in your blood. Blood, when we're looking at needing a transfusion, it's not just as easy as just give them blood. We all know we have blood types, and it can be tricky when looking to have blood donors. So there are genetically determined proteins. Your blood type is genetically determined. What it is there's antigens or there's little keys in your blood that recognizes other outside factors as being foreign. And when those factors are there, your immune system will attack it. For example, if you have type A blood and you're given type B blood, your body will attack it, and you will then begin to have your blood agglutinate or clump up and clot, for lack of a better term. It's kind of like looking at lumpy milk. That is why they look at blood type. What they do is they look at the antibodies that will cause this reaction so that they can pair up what type of blood to give you. There are over 30 red blood cell antigens, but the most vigorous or the most reactive ones are caused by ABO and Rh blood group antigens. That's why you have, say, A positive and A negative. We'll talk about that. Okay, There's two antigens in the blood. There we go. There's two antigens in the blood. You've got A antigens and you've got B antigens. If you've got one or the other dictates what type of blood you will be. If you have none of these antigens or these factors that can trigger this immune response, you would have type O blood. If you have the presence of both of these antigens, then your blood is called AB. So if you have the presence of A antigen, you have type A blood. If you have the presence of B antigen, you have type B blood. If you lack both of the antigens, you have type O. Type AB blood can receive either A, B, AB, or O. They are the universal recipient. They can take anything. They are also fairly rare. If you have type B blood, you can either receive B or O blood and nothing else. A blood, it's the same thing. It's either A and O and nothing else. Type O blood can only receive type O blood. They are the universal donor. They can give blood to anybody. Let's look at this chart. Chart 10 or table 10.3 on page 351 of your book. You've got AB blood group, which has the antigen A and antigen B connected to the red blood cells, so they can receive anything. They can react with A antibodies and B antibodies, so they can take anything. Type B blood only has B antigens on the blood cell, so A antibodies within the blood will react and cause a reaction if they're given type A blood. So they are A, B, and O only. Type A is the opposite. You've got the B antibodies that will react. And then type O. Type O is a universal donor, but they are not a universal receiver, which means they can only receive type O blood or they will have an issue. On top of the blood types A, B, and O, we also have Rh blood groups. They are named because they were originally found in rhesus monkeys. Most Americans are Rh positive. Some people, though, are Rh negative. If you mix the two blood, you will have hemolysis, or you will have a breaking apart of the blood cells. But it doesn't happen immediately. So what happens is, if you've got an Rh negative body that gets Rh positive blood, it's positive because they have one of these 8 antigens that have been found. Okay? If you've got an Rh negative person, that means that they don't have the antibodies for these antigens. So what will happen is, the first time they receive a blood transfusion, you won't have an issue. It's the second time, because then they've developed antibodies to these antigens that they will attack the blood and break up the blood cells. You see this in pregnancy, and it's only an issue when the mother is Rh negative and the father is Rh positive, because Rh positive is a genetically dominant trait, so that the child may possibly inherit the Rh positive factor. What happens is, when the mother gives birth, the first child that is Rh positive, it is not an issue. But there is an intermingling of blood between the child and the mother as the mother gives birth. So what happens is, once the Rh positive blood gets into the mother's bloodstream, she starts to create antibodies. So then what happens is, if she has a second child that is Rh positive, the mother's antibodies can then go and attack the fetus, because the fetus may be Rh positive. RhoGAM can prevent the buildup of the anti-Rh antibodies in the mother's blood. And so the first pregnancy, you'll have no problems, but then the immune system becomes sensitized. It's the way our immunizations work in that they sensitize the body for the foreign object, for the virus, for the bacteria, and then once you're exposed to it again, your immune system attacks it. It is the exact same thing in the pregnancy. This is why blood typing is pretty important. When we look at having to find out what kind of blood type we are, we mix blood samples with an anti-A serum and an anti-B serum. And then it depends on how they react to figure out what the blood type is. So typing for A, B, and O and typing for Rh factors are all done in the same way. You take your sample and you test it with A factor, and you test it with B serum, and you test it with Rh factor to see if there's a reaction within the blood. And here you go. So here's a picture. You've got type A, which has A and B antigens. You will have the agglutination or the clumping of both of those samples. So this would be a type AB. Type B, you see here, you only have the clumping of the type B on the anti-B serum. Here's the anti-A, and then you'll have no reaction at all. And I think we are going to end it here. I'm not going to test you on developmental aspects of the blood, so we are going to move on. And that's the end of the blood chapter. Thank you.